Hey everybody, I'm Dr. Phys, and let's learn some physiology. Alright everybody, we're back. So thanks for being here. Today we're going to talk about an important concept called homeostasis. What it means to have a negative and a positive feedback loop, what it means to have a reflex control pathway, and we're going to go even deeper and explore protein-mediated channels and how they help us maintain homeostasis. So let's get right into it. So you might have heard the term homeostasis before, but just to get everything defined and everything on the clear, the National Institute of Health states that homeostasis is a state of balance among all the body systems needed for the body to survive a function correctly. So in other words, homeostasis is the ability for our body to regulate itself, to keep ourselves at normal speed, and to make sure that we function well and that we can keep going.
Just coughed and farted.
It's sort of like a Tesla auto-driving car that corrects itself when we maybe veer too far into the right lane or the left lane. The AI in the system corrects itself and keeps it going straight. That's exactly what our bodies are doing. An example of homeostasis could be sweating. Let's go to the board. Alright, here's our friend Bob. Bob is receiving a lot of rays from the sun and his skin is overheating. What usually happens when we feel overheated? We sweat, right? So let's talk about that. Bob receives rays from the sun, that overheats his skin, and under the skin, something called a receptor will pick up that signal that the system's being overheated, and it'll send a signal up to the control center through something called the afferent pathway. The control center will hear, hey, we're getting overheated. And it'll say, okay, whoa, we've got to calm down. How can we do that? I'll send a signal over to something called an effector to fix that. So the control pathway sends a signal down the afferent pathway to an effector. An effector is just an organ that's specified to help control a problem in the system. For example, if we're getting overheated, sweat glands would be the effector. Sweat glands in this case would release sweat, cool the skin, allowing the water to evaporate and take the energy with it, and the skin would be a lot more cool. This would help over time to cool down the skin from how hot it is, and thus we've maintained homeostasis. This happens again and again and again until the body is completely cool and we can function at 100%. So for all you math nerds out there, let's put it into a graph. So this graph represents our temperature over a certain amount of time. This dotted line here is the average temperature that our body likes to be at. When discussing homeostasis, every system of the body has a specific point where it likes to stay. This point, in terms of temperature, is 98.6 degrees Fahrenheit. This is called, in general, our set point. The brain controls whatever set point needs to be. This could be different in our kidney, it's different in our skin, it's different in our heart. Every system of the body has a set point. The goal of the body is to maintain that set point. So, for example, when we get overheated, our temperature goes up. What happens? Our control center tells through the afferent pathway to our effectors that we need to cool down. We'll cool down, and then once we stop cooling down, we'll heat back up and try to get back to set point. We'll heat back up. We'll receive a signal that we're getting too hot, and we'll get cooled back down. And it just keeps happening like that, over and over again, in a normal system. This range of normal fluctuation is actually just called normal range. Our body doesn't always stay 98.6 degrees Fahrenheit. It fluctuates, give or take a degree. But this fluctuation is totally normal. We can see here that the body is attempting to get back to set point. Every time it gets too cold, it warms itself back up. It gets too hot, it cools itself back down. So this system of attempting to return to set point is called negative feedback pathways. Negative feedback pathways are super important for our body and is a key element of homeostasis. You might be wondering what happens when we don't go back to set point. Let's talk about that. Say our thermoreceptors or receptors in the skin are broken, Well, what will happen? We won't sense that we're getting overheated, and our body will continue to get overheated. Our control pathway won't know, and we overheat, and we could potentially die. This is called a positive feedback pathway. Positive feedback pathways are when the body does not attempt to go back to set point. This can be on purpose or it can be by accident. It really just depends on the system. But in the case of sweating, we usually try to go back to set point. The same sort of thing happens when we have a fever. Actually, our body will attempt to overheat itself in order to kill all the pathogens within our system, but it usually tends to, once those are done, try to go back to set point. Sometimes our body won't be able to return back to set point because it's so overheated. This is where we'll see fluctuations, attempting to go back to set point, but our temperature keeps getting higher and higher, and that's not good. This is called inadequate negative feedback pathways. So it's inadequate, we're trying to get back to set point, we're trying to be negative, but we can't. And so this is where doctors have to intervene and use drugs and whatnot in order to help our body reset and get back to the set point. In reality, almost every system of the body maintains homeostasis in order to keep its normal function. Let's talk about it on a cellular basis, because this will be the foundation on everything we understand with physiology. Looking at a cell is encapsulated by something called a plasma membrane. That plasma membrane sort of acts like, I guess, our lungs. When we breathe in oxygen. When we breathe out CO2. We sort of allow things to go in and out of our system that way. The same principle applies with plasma membranes, is that plasma membranes are kind of the gate guard or the barrier between the inside of the cell and the outside of the cell, and they determine what goes in and what goes out of the cell. So let's talk about different ways that ions or molecules can go in and out of a cell. So let's dig deeper into the concept of how a plasma membrane lets things in and out of the cell. Starting with something simple. It's actually called simple diffusion. Let's define what diffusion is. So when we spray a perfume, you always hear the word, diffuse. You diffuse perfume across a room. Or you have, an essential oils diffuser. What is that doing? That's taking a concentration of chemicals, spraying it into the air, and allowing it to spread out across the entirety of the room, hopefully being equal. You take that concentration, it's very high, the concentration is super high in one area, and then over time it diffuses, it spreads out across the entirety of the room. The same principle is sort of happening with diffusion on a molecular scale within cells. Molecules will always want to maintain equilibrium. They'll want to move towards an area where there aren't as many molecules. Say you have on the outside of the plasma membrane or the extracellular fluid, you have a bunch of oxygen ions. On the inside you have intracellular fluid. Naturally, these O2s on the outside of the cell are going to want to try to get over here to the ICF or the intracellular fluid. Diffusion rules would have us understand that this is a concentration gradient. It will want to move from high concentration gradient to low concentration gradient in order to make it equal. The plasma membrane will only let certain molecules through, usually non-polar and pretty small molecules, and O2 fits the description. So you'll see O2 begin to move through the center, and now you have that O2 going over here. Oh, and then let's see, 2 and 4. This one will move through the gradient, and then at that point, you have an equal amount of oxygen on both sides, in the intracellular fluid and the extracellular fluid. Thus, O2 has successfully diffused across the membrane in a term that we use called simple diffusion. What if there are other molecules that are in higher concentration outside the cell than inside the cell, or vice versa, but they can't fit through the membrane? They don't meet the membrane's qualifications. What do? Well, that's where proteins come in. Let's cover the different passive channels. So our first passive channel that we discussed is called a leak channel. Leak channels can be specific to certain ions, or they can let in a general amount of ions or exclude certain ions. You have different channels within your neurons, for example, that exclude calcium, and you have different channels within cells that only allow potassium to flow out or in. Leak channels will allow bigger ions to diffuse, so naturally without any energy being used, diffuse across a membrane. Depending on where the concentration is higher, that will depend on where the leak channels allow potassium to flow. So if there are a lot more potassiums out here than in here, then we would expect the potassium to flow where? Potassium would flow from the extracellular fluid into the intracellular fluid. Thus, we would maintain an equilibrium and have the same concentration gradient. Our next passive transport channel is called voltage-gated channels. And to put this one simply, along our plasma membrane, we have something called an electrical gradient. Typically, within the side of the cell, we'll have a negative charge, and outside the cell, we'll have a positive charge. When you have an influx of sodium on the outside, leak channels or ligand-gated channels, which we'll talk about in just a bit, will allow for some sodium to come in. Not very much, but it'll allow for some to come in, and eventually over time they'll start to build up in the ICF, or the intracellular fluid. That will change the electrical gradient, because these are positively charged ions. That'll negate some of the electrical current, and will cause a reaction within the voltage-gated channel. So let's talk about what the voltage-gated channel is. Voltage-gated channels typically are specific. At least the ones that we've talked about thus far. We have potassium-gated voltage-gated channels and sodium-gated, and we have sodium voltage-gated channels. We'll talk about a sodium voltage-gated channel. This channel doesn't require energy, but it does require this electrical gradient. The way this channel is set up, that it has an activation gate at the bottom, it has an inactivation gate, and it has a spring-loaded protein that is controlled by the amount of calcium that's on the outside of the cell. When the cell has enough positive charge on the inside and it's negated the negative charge on the inside of the plasma membrane, that will affect what kind of pull it has on the positively charged calcium ions, thus causing the spring mechanism to rise up and not be weighed down as much. It would pull on this string, we'll call it, and thus open up the gate at the bottom of this voltage-gated channel. Then, you can imagine what will happen. At that point, sodium will see its opportunity and attack, and it'll start flowing into the cell. At which point, the inactivation channel, in order to prevent too much from coming in, will quickly close the voltage-gated sodium channel, and the reaction will continue to happen. This is called a ligand-gated channel. Ligand-gated channels are controlled by something called, as you can probably imagine, ligands. What is a ligand? A ligand is just a molecule or particle of some kind that specifically can activate the receptor that's at the top of the protein channel. So, in this case, if this is a sodium ligand channel, if there's a lot of sodium in the extracellular fluid, sodium will attach to this receptor, and that receptor will tell the protein that it is a-okay to open up its lid, and that gate, that gate on the ligand-gated channel will allow for sodium, other sodium, to come through and begin to flow into the cell by diffusion. Mechanically gated channels are pretty simple. Basically, they involve movement of any kind, really. If there's a movement outside of the channel and it's specific to sodium, say sodium comes up and taps it, hey, I want in. It'll basically run up against the board like this. And until it finds this channel, and then once it taps that, then I'll just draw this. The channel opens and it allows it to flow through by diffusion. Alrighty, so now that we've talked about our channel proteins, let's talk about our carrier proteins. This still falls under the category of passive transport, which means that it doesn't actually require energy and it moves particles from its higher concentration to lower concentrations. It moves particles with its gradient. Carrier channels act as a revolving door. Say this were to be a carrier channel that's specific to sodium. Sodium would come in, attach to the carrier channel. The carrier channel would recognize that this is a sodium ion, and then something that would happen that's called a conformational shift. The entire protein itself will actually shift from being open like this on the outside to open like this on the inside, allowing the sodium ion to flow into the cell. Let's move on to active transporters. So active transporters fundamentally will move ions against their concentration gradient. Let's look at something called a sodium-potassium pump. The sodium-potassium pump is an active transporter that will take calcium from outside of the cell, pump it inside of the cell, and will take sodium that's inside of the cell and pump it out of the cell. This is moving against its concentration gradient. If sodium is getting pumped outside into more sodium, and potassium is getting pumped back into sodium or to potassium. The reason that this is important is that it helps us maintain our electrochemical gradient. The way that this is energized is by a phosphate group. Alrighty. So the last bit of information that we need to cover is our secondary active transport. This doesn't technically involve ATP, but it indirectly uses the electrical gradient that's set up by the sodium-potassium pump to act. We have two different types of secondary active transporters that we need to know of. The first one is called your antiport transporter, and your second one is called your symport transporter. Antiport secondary active transport proteins will take an ion and push it against its gradient, sending it out into the ECF, or more ions. At the same time, that will pull another molecule, gradient X here, will be pulled into the cell against its gradient as well. The same concept happens with symport, but instead of one going one way and one going the other, both will go against their gradients.